We can start if we are ready.
Yes, good afternoon, everyone. I, on behalf of World NCD Federation, welcome you all to the 10th webinar of the International Webinar Series on COVID-19 and NCDs. Today, on the topic dose optimization for NCDs during COVID-19, I welcome all the esteemed speakers and the delegates. The moderator of our session today is Professor Sameer Malhotra from Professor and Head Department of Pharmacology, PGI Chandigarh. Also with us, we have the President of the World NCD Federation, Professor J.S. Thakur. I welcome you all, sir. Today, we have with us 5 speakers who would be taking us to the dose optimization for NCDs during COVID-19, the process of developing evidence-based guidelines with case study, and also promoting physical activity during COVID-19 pandemic. To start the session, I would like to invite our moderator, Professor Sameer Malhotra, to please start the session. Dr. Sameer Malhotra is the professor and head. He is also the member of various professional organizations and executive member of the World NCD Federation. He is the coordinator of the Center of Excellence for Evidence-Based Research in NCDs and LMICs. He is involved in regular conducting of workshops on clinical pharmacology, clinical research, and bioethics, and is also the convener for Institutional Ethics Committee, PGI Chandigarh, and member of Institutional Ethics Committee, Punjab University, Chandigarh. Welcome, sir.
Yes, thanks. Good afternoon. The photo you have shown is, people will think it's another person and the person sitting here is someone else. Anyway, very warm welcome to all of you. So the session today is slightly different from what people in these webinars have been accustomed to, because we will be talking about something not many of you will be familiar with that stuff. For example, when we talk about dose optimization, the first thing that comes to mind if you are a person involved in healthcare, it will be dose optimization of radiation, radiotherapy, or CT scans. But here we are going to talk about drugs. Especially, this COVID has shown that if you had looked at prescriptions during COVID period, the dosing of drug went, haywire. It was pretty bad all over the country, in fact, in many other countries as well. But when we talk about dose optimization for in this particular setting, historically, if you remember the discovery of insulin, we knew that for children, the dose of insulin should be lower. Right now, what we are doing is self-monitoring of blood glucose. Each patient individualizes the insulin dose for himself or herself. But, so that was, we decided that insulin in children, if it has to be given, should be milligram per kg and not a standard dose. That was how we individualized the doses. But then people, anti-cancer drugs were discovered. They were found to be toxic even in those dose ratios. And we decided that they are better dosed on milligram per meter square basis. A next step in dose optimization came when there was enough technology to collect a patient's blood and check the blood levels of the drug, an approach we call therapeutic drug monitoring. Now, if you have studied some during your MBBS period or your PhDs, this is approximately the knowledge about optimization of dose that will be traditionally given in textbooks. But times have changed. Nowadays we use some other methods. Now I understand these methods will be highly difficult for you to understand, being from different backgrounds. Even us who are in that field find them difficult to understand. So I have requested the speakers to keep it as simple as possible. Of course, we have time later on for question answers, and we can discuss if there are any issues. All the speakers have been in this area of dose optimization for many years, although they are from different organizations, but they are working together to improve this and to collaborate, to find out if anything new can be made or how best we can use our resources. And they will talk about it later. I won't sort of jump ahead and tell what they are going to talk about in details. Then we have an expert from the Canadian government who works in the Health Ministry Canada and who has been involved in developing evidence-based guidelines, and even during the COVID period, he's a systematic reviewer and a person very much in demand nowadays because of the way we look at guidelines. And finally, so as after 1.5 hours of sitting, so as to remind you to be physically active, we have another great speaker who was with us till for a few years back, and we miss him, Dr. Sanjay. And he is the professor and head of physical medicine and rehabilitation at AIMS. So I request the first speaker to start the session. Thanks.
Thank you, sir. I would now invite Dr. Nusrat Shafiq, Professor, Department of Pharmacology, PGI Chandigarh.
Good afternoon, everyone. At the outset, let me thank the organizers and at NCD Federation for giving this topic a place in their deliberations. It's as Dr. Samir had already pointed out that this is rather a complicated topic, but it is very important that we give its due credence, because we are talking of innovations these days. We are talking, and especially the current pandemic has made us think about our positions with regards to innovations. So, and especially in the field of therapeutics and prophylaxis. And when we talk of innovations, we cannot just talk about new chemicals, new compounds. We have to be very conversant with the various important issues related to those that could go, that could play a very important role in making a thing work or not work. So having said this, I would like to give you a brief background about how we have planned out this session. Basically, I have with me our two partners who work with us in a consortium for dose optimization. This is a very nascent group. I mean, at best, it can be called as a group only, which operates through a memorandum of understanding. And but there is a lot of work that this group has started. Dr. Rajan, who will be the last speaker here in this session for dose optimization, would be detailing, I'm sorry, I'll stand corrected, not the last speaker, who would be one of the speakers in the session, would be detailing about this group as well. So how we decided to have our schedule was we thought that it would be a good idea to walk you through various steps in the drug development process, wherein and emphasize and give you a brief overview of how we make decisions about doses, and perhaps give you a feel about how the entire dosing business operates in the entire life cycle. Then followed by this would be a session by Dr. Vikram Gota, who would give you some case studies as an example. And Dr. Rajan will touch upon various, how you various training modules which are available, and various kinds of trainings which will help people in our countries, other developing countries, and other areas as well, how they can develop themselves up in this area. So having said this, I'll come to my topic, which is understanding the significance of dose optimization in the life cycle of new therapeutic entity. When I talk of, say, therapeutic entity, we mean both the entities, chemical entities, biological entities, which are used both for therapy and as well as for prevention. Yeah, I hope everybody can see my slides there. Can somebody
Yes,
let me
yes.
Yes, please go.
Thank you. So when we talk about dose optimization and the right doses, one cannot just but ignore the famous saying which was given by a 16th century scientist Paracelsus, who is I'm quite confident that most people would know about him. The important statement that he made was, all things are poison and nothing is without poison. Only the dose makes a thing be poison. I mean, the fancy Latin original is given above. I didn't read it out because I don't think I would be good at reading it out in Latin. So basically, the adage holds true even so many years after his initial pertinent statement. And a lot of what we do in pharmacology revolves around this only, to find the right dose, a dose which will distinguish what is useful from what is not. So when we talk about dose, it is, reflexively what comes to our mind is the amount of the drug or the amount of biological entity, but it goes much beyond that. There are other components of it, the route of administration, the frequency of administration, the duration also, because sometimes it's a cumulative dose which makes a difference. Then it gets a little more nuanced. I mean, what is the dose that for certain conditions you have to give a certain amount up front and then you give a distinct amount later. Typically in pharmacology parlance and in therapeutic parlance, we call it as loading dose and maintenance dose. So basically it is a gamut of all these which are taken together, which is what constitutes a dose. So whenever we are talking about dose in our future discussion, it is this composite that we will have to keep in mind. And then there are other layers which are added to on it. We have to understand with with respect to the timing of administration as well. So the conventionally timing of administration would not come as a part of dose, but it's spacing of a drug administration with respects to food and other drugs that a patient may be taking, especially when we're talking of non-communicable diseases, for which protracted dose administrations and multiple drugs are usually required. Then some things like, then we need to understand a little bit about what is the standard dose and what is individualized dose. Standard dose is when we say there is one common dose which would take care of most of the patients most of the times. Now this would be applicable for a large majority of cases. For example, metoprolol for hypertension. We have a range of doses and we don't usually do weight-based dosing there. Or dosing based on other parameters. But when it comes to certain other drugs and certain other conditions, it would be a weight-based dosing which would become more important, or a dose which is modified based on a creatinine clearance. That is, if there is a renal impairment, the dose may get modified. There are many other factors, and obesity itself may warrant a higher dose. So when we take into consideration these factors, which are called covariates, and we tailor the dose accordingly, then it becomes an individualized dose. So a lot of data needs to be generated for us to say with confidence that we can either have a standard dose or an individualized dose, and this would be the dose. And it works through the entire life cycle of a drug development. So when we start from the very initial phases, when we are doing the in silico molecule searching, or we are doing a high throughput screening to see if the given compound would be effective or not, when we are running in vitro experiments, we need to know how much of the drug we need to put in there to see an effect, to see a response. Then subsequently, when we are somewhat confident about the drug being actually bringing about some kind of desirable response, then we run it through multiple studies, which are typically done in animals and which are called as toxicity studies, where we do it in acute phase and in subacute stages of chronic phases, and we see what are the different doses at which a drug may become harmful or useful, or and what are the doses which are bringing about toxic effects or side effects, and what are the kind of side effects, and we go on accumulating all these data. The purpose of getting all these data is to find out that particular drug, a dose of a drug, of a potentially effective compound, which can be taken further for clinical evaluation. Now here comes a very, very crucial step. From animal studies, we need to move on to what are called exploratory clinical studies. And I mean, we would have collected by now, we would have collected a lot of information about the range of doses which may be potentially useful while avoiding toxicity, but all this data is from animal or models. And now we have to extrapolate this information to human studies. So we carry out what are called exploratory clinical studies, and then we move on to what are called, and here again, we run a range of doses, and we obtain an information about the way the drug is disposed, if we increase the dose, how does the response change? And when we talk about response, we talk both in terms of some markers for efficacy and a lot of information about side effects. And then we again collate this data and come to a conclusion about 2 or 3 doses which we want to take further up. And then finally, when we come to a study wherein we have to actually move on to a large number of patient population, what are typically called confirmatory clinical trials, we would have narrowed down to 1 or at best 2 doses. And we see which is the dose which we are going to use in actual patient population if the drug definitely shows some kind of efficacy. So, but the work doesn't approve. I mean, this confirmatory trial, the work doesn't finish here. The confirmatory trial tested 1 or 2 doses on the basis of which we would get an approval or otherwise for the drug to be marketed or not. The work continues after that, because in confirmatory clinical trials, a lot of special population would not have been evaluated, wherein you would need to understand how a drug works in those with impaired renal functions, for example, those with chronic renal disease, for example, or those with hepatic failure. I mean, we had that kind of situation for remdesivir when we were talking about COVID-19. We had very limited information about the dose that would be most appropriate for patients with hemodialysis. So a lot of work continued subsequently for getting this information. These are called post-marketing studies. So basically, the idea of this particular slide was to acquaint our listeners about the process which goes on for during drug development. And when we talk about drug, we are talking about most small molecules, our conventional kind of drugs, and also about biologics, immunotherapeutics that are now more flavor of the season. At each of this step, we have to make a decision about whether to go ahead or otherwise. And a very important decision when we take a decision to go ahead is the decision whether what is the dose to go ahead with, what would be the doses to go ahead with, because at every step you would try to narrow down to a few limited doses only. And that narrowing down is not arbitrary. It is based on a lot of information that you collect through different experiments that you would have been running. So there are some basic guiding principles at each of those. And these first foremost is that you need to understand how does the drug move once it is administered, how it is moving through the entire body. I mean, and there are very limited ways by which we can get this information. One of the commonest ways that we obtain this information is that we do what is called as, we assess the levels of the drug at different time points after administration from the plasma concentration. We obtain blood samples at different time points, and we assess the profile of the drug through the disposition profile of the drug through its plasma concentration. So that may not be a very good idea. I mean, it may not be very representative of the target site at which the drug may be acting, but that's a very easily accessible compartment from which we can obtain our information. So most of the times where we resort to this option and we get this kind of a curve, which is the basis of a lot of our decision making. And typically we call it as pharmacokinetic study. And the study that we do is a pharmacokinetic study. We derive a lot of parameters from such a graph that we get, that is the concentration time graph that we get. And we couple it with what is called as pharmacodynamic curve. And this basically would tell us at what concentrations, what kind of response that you would get. Here we have, you basically express it mathematically. Not all drugs would have this kind of pharmacodynamic representation. Here, if you see here stands for the concentration of the drug. So basically what you're trying to do is you are trying to correlate the response of a drug with the concentration of the drug that it achieves in the body. And then once you have this information, it is very important to understand, it is not, when we talk about pharmacodynamic, it is not only the efficacy component that we are taking into consideration, it is efficacy and safety both of which have to be taken into consideration. And the entire clinical pharmacology exercise about making a decision about the next dose level revolves around these two graphs. I mean, how does the efficacy change? How does the response change, side effect profile change as the exposure of the drug is changing? And when we're talking about exposure, we are talking about the constant integral of the concentration time curve, which we call as area under exposure. So basically the entire judgment revolves around the understanding of these curves, and these curves may acquire different shapes for different drugs and different conditions. So what are the non-clinical studies and that we typically carry out? These are the studies that we carry out. I've touched upon these: acute and chronic toxicity studies, pharmacodynamic studies, mechanistic studies, animal pharmacokinetic, pharmacodynamic studies, several in vitro studies. All these studies will help us understand certain levels. So we have some levels which we want to know. We want to know the level at which there is no observed adverse effect. We would also want to know the level at which we see some kind of pharmacologically active action occurring. And then we want to perhaps see at which dose there is a toxicity of 10% of animals, at which dose it would be a toxicity of 50% animals. And then we pull in all this information to come to a decision about the level of dose which would be experimented with in the first time when we go and experiment it in healthy volunteers or in certain situation patient population. So basically there are two approaches that we use. One is based on the no observed adverse effect level that we would have obtained from animals, and the other would be applicable to biologics that we usually use, which is your minimum anticipated effect level. No observed adverse effect level information is derived from the chronic toxicity or subchronic toxicity studies that we carry out in animals. This typically we would be seeing many dose levels, and then we see that dose at which basically there are no tangible adverse effects. And response would be obtained from in vitro experiments usually, wherein you will see we will carry various kind of biomarker based experiments and come to a concentration of a drug uh, from which you will calculate the dose which can be taken safely in human studies. So various algorithms have been devised. One of the common algorithms that uses Noel approach is the one which is given by US FDA, wherein once you have a Noel effect, you carry Noel dose level, you obtain a human equivalent dose, you apply a safety factor just to be safe, that you don't want to start with a very high dose in healthy volunteers and land up in trouble. So this is one of the approaches which is used. The other approach that is used is the Mabel approach. As I said, herein you would run a gamut of assays, and you see which is the assay at which you get to one of the important responses and at the least concentration. So for example, here is a dose which was a drug, which was a biologic. I won't go into the details of the mechanism of action, but it was acting through various molecular path mechanisms. And these are listed here. It was acting through interferon gamma pathway, interleukin 6, etc. It caused T cell activation, etc. So this range of assays were experimented upon. And what was realized, that tumor cell cytotoxicity occurred at the lowest concentration. The concentration at which 20% of response for this parameter was obtained was 1.34. And then this was this information was extrapolated to obtain a corresponding dose for a 70 kg person. Then this dose milligram per kg was calculated. So the dose amounted to 57.4 milligram per kg. Now this is an example of Mabel approach. A similar experiment was for the same drug. An experiment was carried out in animal models, and the dose was again calculated, and it was noted that the dose that was calculated was 0.6 milligram of drug per kg. Now, if you compare this dose with 57.4 nanogram per kg dose, you see a difference of 1,004. So by mistake, if somebody starts with a higher dose in healthy volunteer and with an effect occurring at a cellular level, which can have devastating effects, you could land up in trouble, and the drug may get killed simply because of the side effects it would have produced in a healthy volunteer study. So this was just to give an example of how crucial this important step is. So once you have this information, you carry out healthy volunteer studies. But if the drug has a very high cytotoxicity profile, you may even carry it out in, for example, your cancer chemotherapeutics. You may carry it out in further evaluation in patient studies. What you do is you evaluate several doses of the drugs, starting with your first in human dose that you would have calculated, both when administered as a single dose or when it would be administered as multiple doses. Here I show you graphs from a PCSK9 inhibitor, which is used for familial hypercholesterolemia. And you see how different doses have been evaluated. So what you do not only obtain is the PK profile, you also get what is called as the response in patients. You can see the LDL decrease in the LDL levels at different time points is noted. What is not shown here is also the side effect profile which you would have got at different dose levels. So all these three data sets are taken together and you make a decision about the the dose to proceed within the phase 2 study or further phase 3 studies. So once you have evaluated in phase 3 studies, you would have got marketing authorization if your drug was proven to be sufficiently safe and effective at a given dose. But the work wouldn't stop there. As I said already, there are several patient populations where you would have to carry out further studies in. And the kind of studies you do is, and Dr. Gota would be talking more about these as an example, your population pharmacokinetic studies, PK/PD studies. You may even would like to obtain pharmacogenomic data to see if there are certain set of patients where a lower or a higher dose may be required. We now have certain groups of drugs for which it has become a very important consideration. And then you can come up with the need or otherwise for individualized or tailored dose. Here are some of the very good examples of studies which have been carried out to answer very important question. For example, I'll just see this first one here, where it was understood whether a lower dose of folic acid, if given for that is 10 milligram per week, may be very important in handling methotrexate toxicity than the conventionally used regimen of 30 milligram per week for prevention of infection of toxicity due to methotrexate. When we talk about COVID-19, there have been a lot of dose-related issues, but I think I would like to touch upon just these two issues. Corticosteroids have been a very, very important issue too, and the dosing, the right dose for corticosteroids has been a point of discussion right from the beginning. It was this remarkable analysis which was carried out by Kumar et al., where it was shown that if probably more than 40 milligram equivalent dose of methylprednisolone may perhaps be causing more toxicity than benefit. And subsequently, there was a lot of advocacy for a lower dose of corticosteroids. Then based on post-marketing studies and certain other studies, for your monoclonal antibodies, an approved dose of 2500 milligram was reduced to 1200 milligram. Not only that, the route of administration was also made more convenient from IV to subcutaneous dose. And that was from, if we think of patient and administrator and a clinician's perspective, this becomes a very, very important change in the management of patients because it makes something more convenient, and what you can achieve with a lower dose by avoiding toxicity and achieving good enough efficacy, I think is something that everybody would want to aim for. So that would be all from my side. I think let me know if we can have questions right now or we are going to have it at the end. Thank you.
We'll have at the end, all questions at the end.
Thank you, sir. Thank you.
Thank you, Dr. Nusrat, for giving such a detailed presentation on the significance of dose optimization. For our next topic, I would like to invite Dr. Vikram Gota, who's the Professor, Department of Clinical Pharmacology, Advanced Center for Treatment, Research and Education in Cancer, Tata Memorial Center, Mumbai. Welcome, Dr. Vikram.
Yeah, thank you very much. At the outset, I would like to thank Dr. Samir Malhotra for extending this invitation to me, and also thanks to Dr. Nusrat Shafiq for inviting me for this webinar. So I'll be this afternoon. I've been also talk about dose optimization, significance for practice related issues, questions. So I'll be doing exactly the same. So I'll be talking about what is the significance of dose optimization as far as our practice is concerned. Okay, so I'm having some difficulty in moving the slides. Is there? Okay, I think there is some problem. Okay. Yeah. So now, why optimization? Of course, this is a very, very cliched question and a cliched answer also. One size doesn't fit all, and we know that. I mean, in the sense, and then we have shoes of different shapes here, and obviously, an adult cannot fit into the shoes which is in the middle of the frame, and likewise for other age groups as well. But then, what substitutes for shoes in a real life situation for a drug is ethnicity, pharmacogenetics, tolerance for adverse events. Like, for example, it is historically believed, and and there are several studies to support this belief that the tolerance to chemotherapy, at least in the case of cancer, in Indian patients is less as compared to other ethnicities. And in local prescribing practice, depending on how many how drugs are prescribed, polypharmacy practices, and rational prescribing practices, and so on and so forth. So that may also, because of polypharmacy, can affect the kinetics of drugs in several ways, and that one should be aware of. And the quality of drugs itself is an issue. Like, for example, if I'm told that this is a size 10 shoe and it should fit me, and I buy it from Amazon, and if it happens to be a size 6 shoe, so obviously it doesn't fit me. So that is the quality of drugs is something like that, where they claim that it is 100 milligrams, but the drug happens to have just about 60 milligrams of active principle. So those are some of the issues which are very unique to some of the LMICs, low and middle income countries. And as well as why one size doesn't fit all, because there are so many variations based on age, sex, and so on and so forth, which has their own idiosyncrasies as far as the disposition of drug is concerned. So that is the reason why we need to optimize. So now one of the so I'll speak from our own example. I mean, just so that I can tell you how we used optimization using pharmacometric methods in our own population. So one of the important questions that was kind of plaguing the minds of our oncologists not too long ago was, suppose a woman of early breast cancer is overweight and obese. Okay, so should we be dosing the woman as per her actual body weight, or whether we should use any alternative descriptors of weight? Sorry. So whether we should use any alternative descriptors of weight, like lean body weight or something like that, to dose. And the reason why this question became important back then was because a woman who's overweight and obese, and she has a body surface area of about, let's say, 2.2 meters square. So paclitaxel is used at a dose of 175 milligrams per meter square. So 2.2 would translate into a dose of about 350 milligrams. And generally, it was believed that paclitaxel in anybody, if it is used beyond a dose of 300, a certain dose can cause toxicity. So there was a lot of uncertainty as to whether women who are overweight and obese should be dosed as per their actual body weight, or we should calculate the lean body weight and then calculate the dose of paclitaxel according to that. So, but the practice back then in our hospital was to dose all women irrespective of their body surface area as per the actual body surface area calculated using actual body weight. So therefore, we went ahead and said, okay, let us do the pharmacokinetics and then see whether the kinetics is any different in overweight women versus normal women with normal body mass index. So that's how what resulted in this study, and then we enrolled about 36 women, and some of them were in the overweight obese range, that is BMI more than 25, and half of them were in the normal BMI group, that is less than 25. And so we had some very obese women also with BMI going up to 34 and so on. And when we did the pharmacokinetics, what we noticed was that there was no difference. So all women were dosed 170 milligram per meter square, irrespective of what the body surface area was. And the body surface area was calculated using actual body weight, not any ideal body weight or whatever. Okay, and what we observed was when we dose according to that practice, there was absolutely no difference in exposure to paclitaxel. You can see that the line is going flat. I mean, in sense, there's absolutely no difference in exposure to paclitaxel in an 18-year-old, in a woman with a body mass index of 20 versus a woman with body mass index of 34. So there was no change. So therefore, we could conclusively prove in a very small study of 36 patients, we could conclusively prove that there is no significant difference in paclitaxel exposure between normal and overweight women when dosed as per their actual body weight. So now this became a standard practice in our. So it was earlier also standard practice, but now they decided that there's no need to change the practice because we now have proof from a pharmacokinetic study that the exposure doesn't vary, with body surface area. Now coming to the next problem of quality of generics. So a study which we published in the British Journal of Clinical Pharmacology was about comparison of asparaginase activity in generic formulations of asparaginase, in vitro and retrospective analysis of asparaginase monitoring in pediatric patients with leukemia. So we must know that asparaginase is a very important drug for the treatment of acute lymphoblastic leukemia. And it has been practice, standard practice to use asparaginase that was available, and there are about 10 odd brands of asparaginase that are available in Mumbai. And our pharmacy, based on convenience, would stock different brands at different points in time. But we started kind of monitoring these levels because we were a little suspicious as to whether the levels are actually good or not so good and so on. So fortunately, we had a few patients who had received the innovator also, but off late, innovator was no longer available. And what we did was, all patients who received the generics, about 200 patients, 200 plus patients, we measured the trough asparaginase activity. And what we observed was, when you look at the trough asparaginase activity, and then, there is enough evidence to show that the trough asparaginase activity should be at least above 100 for the drug to be very effective. But of course, more recent literature showed that, even the activity of 50 is good enough. But what we observed, to our utter surprise and shock, is that, majority of generic drugs had trough asparaginase activity less than 50 international units per liter when it was administered to children at the standard dose of 10,000 international units per meter square. Okay, that was the dose that was used, and then majority of children had low exposure to asparaginase with the generic drugs, but a few children of earlier times who managed to get the innovator had adequate levels. Okay, so now this raised several questions as to what is the ideal way to dose a leukemic child with a suboptimal quality drug. I mean, in a sense, we don't have good quality. So now, if you have to make do with whatever we have because we don't have an innovator in our country, then what is the dose that we can? Then we deviate from the standard dosing regimen. So the standard dosing regimen is 10,000 international units every third day. Can we deviate? Can we give 10,000 international units every second day, or something like that? Huh? So if we can deviate, what should be the dose and what should be the frequency of administration? So this is a question that we could not, although we wanted to, we could not answer it because thankfully by the time this paper got published, the PEG asparaginase had come into the market. The generic PEG asparaginase was available. We had done studies with generic PEG asparaginase and shown that it has comparable activity as compared to the innovator. And slowly but steadily, almost all the oncologists in Tata Hospital had weaned away from the native asparaginase to the pegylated version of asparaginase, which was giving adequate levels. So they could not answer this question. But incidentally, some of our colleagues in Kolkata, in Tata Memorial Center, Kolkata, who are also using asparaginase of the native asparaginase from E. coli, they also face similar problems. And then they did some pharmacokinetic studies, and then they could optimize the dose and said that they identified one molecule and then said if you're using this product of asparaginase, then instead of using 10,000 international units every third day, if you use 10,000 international units every second day, you will be able to manage adequate activity of asparaginase in plasma, and thus they could change their practice based on pharmacokinetic studies. So this is the power of pharmacokinetics, because when drugs are developed, most of the development work happens in Western countries. There is very few data in from Indian population. And even so, when the drug is out of patent and generics are available, the quality of generics can be suspect sometimes, and therefore, pharmacokinetic studies are necessary to understand what is the exposure you are getting with the generic drugs. And then, if the exposure is less, it should not happen on a regular basis, hopefully. But then, whether we can devise alternative dosing strategies so that, we match the concentrations that you would otherwise achieved from the innovators. So this is just one example. The other example is more pertinent for which I gave about paclitaxel, because that is actually classical pharmacometrics where you're not suspecting the drug, but it's just that the dispersion kinetics in different populations is what is being questioned, and where you're trying to match the exposure. So those are the more obvious uses of pharmacometrics. So now, having discussed the power of pharmacokinetics and pharmacometrics, especially the population pharmacokinetic study that I alluded to in the paclitaxel example in early breast cancer. So what are the practice-related questions during COVID? So some of the practice-related questions in COVID is timing of chemotherapy. Like for example, there are so many waves going. We know that when the wave is at a peak, the incidence of COVID is also very high. And the other time the incidence of COVID is low. So, when can you give chemotherapy so that, they don't kind of catch the wave in a bad way? Okay. And what should be the dose of the standard chemotherapy at the time of COVID? Can you use a full dose, risk myelotoxicity, and then therefore the risk of COVID? Do you have to use modified doses? And if you use modified doses, modified means obviously lower dose, and then you are going to risk recurrence in a curative cancer. Or what are the quality of life issues in a metastatic disease? So whether are you willing to compromise the dose in a metastatic disease, because anyway that it is not curable. Therefore, at least you don't want the patient to have such bad myelosuppression that they end up contracting COVID and suffering as a result of that. So these are some of the issues that kind of are very that arise very commonly during this epidemic. And in a sense, per se, we have not. I'm not trying to address any of these questions ourselves, but I'm sure there are several pharmacokinetic studies that might have happened in this period to look at how to kind of manage the neutrophil nadir, and with regards to a full wave of COVID and so on and so forth. I'm sure there must be a lot of studies, especially in metastatic setting, as to how low can we go so that we don't compromise on the quality of life. We don't compromise on the myelotox we don't compromise on the risk of COVID. At the same time, we keep the neutrophil count at a slightly variable level. And at the same time, you don't harm the patient too much by reducing the dose, so they have a early relapse of the disease. So those are some of the fine balancing act that would be very, very good questions for any pharmacology lab to answer in the time of COVID. But we are answering a slightly different question at the time of COVID. Okay, so as to this question is related to vaccine. Like for example, if I have to, if there is a patient of COVID, who is on, let's say, cancer treatment, what happens to the vaccine titers? Okay, so what are the antibody titers following vaccination in a cancer patient with COVID? So that is a study which is actually ongoing in our center, but we are not directly answering any question, at least in my department, as to what should be the dose of chemotherapy or the timing of chemotherapy, and or the timing of vaccination with respect to chemotherapy. Those are some of the very interesting questions that could have been addressed pharmacokinetically, and I'm sure there are several groups doing that. But as far as our group is concerned, we are actually looking at antibody titers in COVID patients who are also our cancer patients, and trying to see whether the antibody titers are kind of comparable with the normal population, or is it low, or is it high, or whatever. Okay, So now, if you have a research question that is affecting your practice, something like dosing or whatever, how do you address that question? So, like Dr. Nusrat very clearly alluded to, you can do population pharmacokinetic studies, you can do some PKPD modeling, especially if you have a robust pharmacodynamic marker. So you can do PKPD modeling and so on and so forth. So that will help you to address the question as to what is the right dose to use. But how do we do it? I mean, do we have the infrastructure? Do we have the capacity, and so on and so forth? So with those, when we face those questions, we came up with a very ingenious answer, and that was to collaborate. Because we know that certain centers have certain expertise. Like for example, we as a Tata Memorial Center have very good expertise in clinical pharmacology, therapeutic monitoring, and we have a very good bioanalytical setup. So we have my colleague Dr. Rajan in Manipal College of Pharmaceutical Sciences, who is a very good pharmacometrician. He can do the PKPD modeling. And we have Dr. Nusrat in PGHM Chandigarh, who has some fantastic research questions. In fact, the reason why we started CODE was because she was grappling with the question on what is the right way to dose vancomycin in ICU patients. Okay, so that was the problem that she came to us with, and that is when we decided that, okay, if we get together and kind of design studies together, based on our mutual expertise, we will be able to kind of make contributions to which matches our strength, so that the overall quality of studies can get better. So that way, many times it is so happening now that the questions are coming from PJ Chandigarh. We are able to give some assistance in terms of bioanalysis using HPLC, LCMS, and so on. And then Dr. Rajan is able to do is able to model the data. So that is how, with these intentions and background, the Center for Consortium for Dose Optimization was born on 7th December 2020, and the objectives are very simple. So we have in a very pilot scale, we one-to-one we were working, and then we could sort out a few population pharmacokinetic studies. Now the question was whether we can this arrangement is sustainable and we can scale it up to include several centers across the country. So can we build capacity for pharmacometrics? And that includes extensive training, organizing webinars, and workshops, and so on and so forth. So that is one of the important objectives. And to create a platform for crosstalk and collaboration between the participating centers to carry out multi-center studies. So, and these are some of the very good examples that, like for example, the breast cancer question that I brought up, or the timing of vaccination in the cancer patient question that I brought up, those could be very elaborately and beautifully answered through collaborative research. So the idea was to have multiple centers kind of come together, brainstorm, identify research questions and come up with appropriate solutions. And more importantly, to facilitate free movement of biological samples and data from one center to another, because that is an impediment actually for any collaborative research, because you cannot transfer your data, your IRB asks you 100 questions, you cannot transfer your samples, somebody will ask you 100 questions. So that is the reason why we wanted to kind of break those barriers, so that we have an MOU in place, so that all these things are very easily possible, and we're able to raise some important questions and answer them. And the strength of CODE lies the fact that we are two multi-specialty referral centers in Chandigarh and NCOPS, and we at ACTREC are cancer specialty hospital, which form the consortium, approximately 3,000 beds between the three centers, and there is opportunity to scale up. I mean, India being a vast country as it is, and with so many regional cancer centers, 25 regional centers, we have an opportunity to scale up the CODE to 25 regional centers, about 15 aims that are already existing or coming up in different parts of the country, and several standalone postgraduate institutes of medical sciences. Okay, because we have several dosing problems that are unique to our population, like the practices are also very vague, sometimes irrational. There are polypharmacy, drug-drug interactions, infectious diseases are a problem. So AKT, anti-tuberculosis drugs. So how do they interact with other drugs? There are so many questions that has to be. Malnutrition in pediatrics, that is another big problem. Our lab is extensively working on malnutrition in pediatrics and how to optimize dose of chemotherapy drugs in malnourished children. especially doxorubicin for that matter, because it is known that the severity of cardiac toxicity increases in children who are poorly nourished. So, so these are some of the questions that we are trying to answer. Okay, so that is how we expect that code will be able to answer some of the problems clinical related to clinical practice that could be easily answered through very well designed pharmacokinetics and population pharmacokinetics studies. Okay, so thank you very much for your patience, and I'll be happy to take questions anytime, either now or later. Thank you.
Thank you, Dr. Vikram, for taking us through the significance for practice-related questions and dose optimization. For the next topic on resource for enabling dose optimization studies and training, I would like to invite Dr. Surulevel Rajan, who would be speaking on the topic and who is an associate professor in the Department of Pharmacy Practice, Manipal College of Pharmaceutical Sciences, Mahe, India. Welcome, Dr. Surulevel.
Thank you. Thanks for that introduction, and I thank organizers for giving me this opportunity to interact with you. As both the speakers have mentioned, so we are trying to focus on how to develop this kind of collaboration and train the manpower in our setting. In our setting, and hopefully conduct similar studies across the country, and kind of meaningful recommendations that will help us to dose optimize in our setting. The catchword is pharmacokinetics. We are familiar with this. But the second word has been used is population pharmacokinetics. That is, it's kind of, I would say, global level. It is 15, 20 years old terminology. The population pharmacokinetics. The current terminology we use is pharmacometrics. So when it comes to pharmacometrics, it encompasses the population pharmacokinetics, pharmacokinetic dynamic modeling, and also the study of basically the drug dose related issues and responses together in a mathematical, rather very quantitative, I would say, not necessarily mathematical, it's a quantitative framework. That's where the pharmacometrics stands here. And there is a division of pharmacometrics at the US FDA, which was spearheading these guidelines, how to do pharmacometrics. And now the other agencies like European, Japanese, Chinese, global, all regulatory agencies have the pharmacometrics divisions in their regulatory approval, which is the clinical pharmacology umbrella. And they give guidelines and number of, there is no product, I would say no drug product for which the pharmacometric analysis has not been done, at least in the recent few years. So that the science has gone that far, and in terms of our country, the training is like training opportunities are less, and broadly I give the picture of where the training opportunities are concentrated and how to use the opportunities for us, at least those who are in other part of the world where the opportunity is not available. So when it comes to academics, and it's always US and Europe. US certain university of Pittsburgh, Maryland, and Buffalo, they are kind of old established universities, and they give kind of masters and even PhD in pharmacometrics and personalized drug therapy or in their system pharmacology. These are all various subdivisions which are growing that use of pharmacometrics in system pharmacology or quantitative pharmacology. Another one is pharmacometrics used for the drug development and clinical applications. And there are pharmacometric applications which are focused on personalized therapy. And there are some short-term courses available at the University of North Texas and Uppsala University, which is in Europe. They give a lot of training at the master's and PhD level. And there is a consortium in Europe. Okay, these are all like bigger universities and training opportunities. But why I'm telling is there are a number of students from our country itself, or in India or other parts of the Asia, have moved to many of these universities, and they are pursuing their PhD or even completed and become pharmacometricians That's why I'm just giving out those names, even though they are not easily accessible. PhD level programs globally, many universities, including in India, at least few universities are giving. But master's level, it's not formally available anywhere across Asia, including India. And sometimes some in medical courses, this has been taught. So then how the training for everybody except a few courses, what are the training opportunities for others? Usually for many of the willing aspirants, the approach is always the conference. Many of the conferences, they conduct workshops on a routine basis, annual basis. So the main conferences will derive the knowledge, because already clinical pharmacology knowledge will be there broadly, but we have to transform that knowledge and add the additional knowledge of pharmacometrics. So the page Europe, Population Approach Group Europe, is the main group in Europe, which is actually kind of a pioneer, I would say, is training the people in pharmacometrics. Then American Conference of Pharmacometrics, Australian group, Japanese group, Chinese group, like that. Even there is one Asian group is also there. And as far as India concerned, we have a group called Population Approach Group India, and now that has been transformed into Society of Pharmacometrics and Health Analytics. For the past 1 decade, they are conducting workshops using for this population pharmacokinetic model. You may think, what is why I'm talking this specifically? One of the questions Dr. Vikram was mentioning about the obesity and dosing of paclitaxel, they've used the population pharmacokinetic analysis using a software called non-null. He has mentioned that particular name of the software or approach also. So that training is very essential for carrying out all this advanced pharmacokinetic and pharmacodynamic analysis. It's kind of evolved from just therapeutic drug monitoring, looking at the drug concentration and making some clinical decision. From there, it has been moved to actually taking decisions and conducting this analysis and come out with this kind of very focused recommendations. And this now code consortium has been started, and you can keep looking at this space of code consortium. Website information will be available, and there are courses and trainings and webinars and training programs have been planned. So these SOFAs and code consortium are the organization which are dedicated for training clinicians, pharmacists, and other healthcare providers who is focused on this discipline in the area of pharmacometrics. So that is the web address of the sofas.net. In their resources page, they have information about the other global professional societies. Training materials are already available. You can find a link there. And pharmacometric tools, what are the tools available and how to get that, and pharmacometric journals where you can refer on all this domain. So all of this information is available in the website of sofas.net. You can access all of these resources, and it's available for anyone, for a beginner to start. And structured training programs are in the offing. Last year it was started, and this year in the coming months, and at least in the coming few months, we are expecting training programs from both SOFAs and as well as the code consortium. So for a beginner, what are the resources we can at least to start, we can go through and understand and start training ourselves. American College of Clinical Pharmacology, in their website, they have pharmacometric resources, ACCP website. You can go, and the University of Maryland has provided the resources for this American College of Clinical Pharmacology. There, this information available. One professor on clinical pharmacology, Professor Nick Halford, that's in the New Zealand, Auckland. There, the pharmacometric, lot of material from the basic pharmacokinetics, population pharmacokinetic, pharmacometric, that kind of the whole level of training materials are available for free. And Monolix is one of the software, which is again, it's free for academics. Their website is elixsoft.com. In their website, they have a lot of training videos and materials and how to get this software for for our purpose, especially academics and clinical persons can use it for free. So this is another important link. We can use it. Next one is a new tool which was available. At least I would say within 1 year it has been released. Pharmaceutical modeling simulation tool called PUMAS. And they have their own tutorial with a lot of case studies, at least 50 pharmacokinetic studies, case studies, and how to, many of this monoliths and pumas and many of the tools, there will be some level of code writing will be there. And the training for write the code and analyze the data, all that information is available in their website, pumas.ai website tutorial page, all the case studies and how to use the tool, all this information is available. So anybody who is interested in this area can at least look in this, know the names, American College of Clinical Pharmacology, Nick Harford's website, Monolix tool, and Pumas tool. There are other tools, actually non-mem Phoenix, there are other tools, but they are proprietary, but it is not available free for training or working. That's why I'm not given that link, but at least this Monolix and Pumas, it is free for academics. And this pharmaceutical modeling and simulation, it is there in cloud. You don't have to even download into computer. You can log into the cloud and you can start working. So these are all the kind of resources which is available for training. Apart from, if you have to carefully look in those two spaces, I would say, SOFAS, Society of Pharmacometrics and Health Analytics India, and Code Consortium India. These organizations, I'm part of both of these organizations along with collaborators. So both of these organizations are coming up with the training programs and webinars in these coming months. And anybody is interested, please keep checking in this space. So thanks for the opportunity, and I would take the questions later. I think other speakers are there, and thanks for the opportunity. Thank you.
Thank you, Dr. Surulivil, for your presentation. Next, we have I'd like to welcome Dr. Avtar Lal. He will be speaking on the topic process of developing evidence-based guidelines. Dr. Avtar Lal is guidelines developer and a systematic reviewer from Canada. Welcome, Dr. Avtar.
Thank you very much, Ria. Let me share the slides. So, can you see the slides, or
Yes.
should I move in a different mode? I can start with this one.
So we can see the slide.
Okay, great. So I'll start with this one. Yeah, good. So I'm starting with the process or development of evidence-based clinical practice guidelines. My task has been simplified by previous speakers. So this is a bit different than dose finding and dose calculation. This is a bit simple as compared to the previous calculations. So guidelines are basically, these are systematically developed statements, and these are designed to help administrator, practitioner, and patients make decisions about appropriate healthcare for specific clinical circumstances. This is very, very important. And guidelines are basically two types. One is consensus-based, and other are evidence-based. So consensus-based previously used the panel of experts or leader used to get together, and they just thought that we should develop some guideline also. And then they used to review a cherry-pick evidence, and they used to prepare recommendation based on consensus only. But nowadays, this is a bit different, that people are more trusting on evidence-based guideline, and this is supported by the best available evidence. And everyone knows about the hierarchy of evidence. And the previous speaker also mentioned that there are evidence which comes from case reports, case series, and other studies also. So the topmost evidence is the systematic review of good quality randomized control trial. Then further comes a randomized control trial, cohort study, case control study. Everyone knows about this thing. Basically, the quality of evidence increases if we go from bottom to top. So in the evidence-based guidelines, basically follow the unbiased and transparent process of systematically reviewing and appraising the best clinical evidence of highest quality. So clinical evidence is there, which is best, which is randomized control trial, but at the same time should be highest quality. That the randomized control trial we're going to include in the system in the systematic review and guidelines should be of no risk of bias. So should be a minimal risk of bias. And these guidelines, well, the opinions are markedly reduced, and these are very helpful for the patients. So in certain cases, the evidence is missing. So then we have to go for the consensus only. So consensus-based guideline and evidence-based guideline, they complement each other. So in the steps of development of guidelines, we basically select a topic, then submit to a guideline development committee. Then the committee will review the topic. Then based on that, they decide we should go for guideline and look for chair, co-chair, and working content experts and methodologists also. And basically a team is prepared who can develop guideline. And there are different tools for grading the recommendation which are being to be included in the guideline. And the commonest tool which is being followed all over the world these days, widely adopted, is GRADE. This is grading or recommendation assessment, development, and evaluation. So in development of guideline, for the first step, which is perform a search of a literature, then this is same literature which is used for conducting a systematic review. And we look for systematic review, randomized control trial, cohort study, other study also if you want. And we search for different databases, not rely on one database. And we prepare inclusion criteria, and exclusion criteria means what type of patients we want to include, what type of intervention. Suppose that we want to develop guidelines for the patient of COVID also. And we need to decide in the beginning whether we want to take adult patient or children, and what type of comparison or intervention we want to see, whether what drug we want to see, what dose we want to see, and so on. And how this is going to compared with nothing or compared with some other drug also. And what outcome we're going to study in this guideline also, that whether we look for mortality or look for other outcomes also. And as far as possible, we should try to look for clinical oriented outcome than surrogate outcome, but both are important. And the design of the study, we have to decide in the beginning whether we want to rely on the randomized control trial or cohort study or case controlled study, or maybe we're going further lower because we have nothing there, no RCT, no other study also. We have to go a little bit lower to case reports even. This all depends upon the availability of evidence. So this is a common methodology which is being followed for systematic review. The same is for guideline also, that we perform a search of literature, then perform the first level selection based on title and abstract. And based on the inclusion criteria, some articles are included if the articles are having clearly mentioning of all the component of PICOs, or these are excluded if these components are not mentioned. And once the first level selection is done, then we go for the second level, which is based on the full text screening of those articles. Articles are included if these follow the PICO or PICO is mentioned in those one. These are excluded if these PICO component, these are not mentioned over there. And here we have to identify the reason for exclusion. So after conducting, selecting the study, we perform data extraction. And for data extraction, we also try to perform meta-analysis. So meta-analysis is very crucial, that if more than one study is estimating the same fact, we can perform a meta-analysis, but the study characteristics should be similar and outcome measured in the similar way. And we should pool like with like, and we shouldn't mix with apple with oranges. So this is basically a meta-analysis. My next speaker will be interested in what is activity or exercise and what is rest. This is basically a meta-analysis comparing the role of exercise, but this is not in patient with COVID. This is other disease also. This was concussion, and comparing the role of exercise compared to rest, and this is decrease in the hospital stay. So in grading the evidence, we look for different components which are very crucial for the development of guideline. One of the commonest and the first most is study design. So we should try to look for a randomized control trial. If we don't have a randomized control trial, we can go a bit lower, look for observational studies also, cohort study, case control study. And the second component, which is very, very important, that we should look for risk of bias, means what is the methodological quality of these studies, or randomized control trial, or observational studies also, and how there is inconsistency, and how the indirectness is there, and how the imprecision is there, and what are other considerations we're going to look for in development of guidelines for any disease, maybe COVID, maybe non-COVID also. So in the meta-analysis, I mentioned already that we look for RCT or observation study. In the risk of bias, there are different tools available for risk of bias. For randomized control trial, people commonly follow the Cochrane, and for observation study, the different other tools are available, Cochrane and other also. We can follow any tool which is a good one. Then for inconsistency, so this is very, very important that in the meta-analysis, there could be, if you want to perform meta-analysis, then inconsistency should be decreased as far as possible, that this could come from the clinical, that the PICO is being considered in meta-analysis, and same thing is followed in the development of guidelines. So we have to look for the same type of patient, same type of intervention, same type of comparison, and outcome measured similar way, and the design of the study is same. And this could be statistical also. This we could look from the forest plot that look for I-square value, and we look for p-value also of heterogeneity. In the grading of evidence, the indirectness is very, very important. If we want to develop guideline in patient with on COVID in children, then we cannot get the data of adult patients. The same way, the dose is very, very important, as previously the author mentioned also. Here also the development of guideline, if you want to develop, suppose this is the guideline for vaccination, then so whether we want to look for 2 doses or 3 doses, which vaccination need to compared with nothing, or compared with one vaccination versus other vaccination also. This all depends. And for grading of the evidence, the same, the imprecision is there. So this tells about the sample size, the confidence interval. If the confidence interval are very wide, then we get a little bit lower in the grading also. And these are very compact, very concise also, then this is very good for development of guideline. Then there are other component we look for development of guideline, which is the publication bias. This is being commonly followed in systematic review and meta-analysis, that if more than 10 studies are there, we can look for funnel plot, and there are other statistical method available for looking for publication bias. And we also look for effect size, whether the study or the meta-analysis mentioned about the dose effect relationship or prognostic factor being considered. So all these components are considered in development of guidelines. And based on type of study, design of the study, risk of bias, indirectness, inconsistency, and other component, we generate grade. And if the studies are good, then we get a very high grade, and this is low, moderate, very low also. And after generating the grade, a guideline committee consider this evidence and consider the values and preference of this intervention, these type of patient, then they develop a recommendation. So the strong recommendation comes from the strong method with the large effect and very precise estimate and fewer adverse effect, whereas the weak recommendation come from weak method, small effect, imprecise estimate, and many adverse effect. And the recommendations, the wording of recommendation is very, very important. If the grading is showing higher moderate effect, then the recommendation wording is we recommend something. We recommend the intervention so and so in patient with COVID, or for recommend vaccination, this type of vaccination in patient with the COVID. If the evidence is low or very low, then we cannot recommend it. We basically can suggest it. So recommendation wording is that we suggest that vaccine so and so should be used in patient with these type of diseases, and so on. So I didn't pick any type of, don't want to be biased that this is basically how we can develop guideline in patient with COVID also. Then this is, we should have question. So should vaccination with so and so be recommended for patients so and so? So for that, we perform a search of literature, then perform inclusion, exclusion criteria, conduct systematic review, and look for population age also. Where if we want to develop guideline in children, then get the data of children only, and look for intervention, 2 doses only, and comparison with no vaccine, and outcome, what outcome we going to consider, or symptomatic or confirmed cases of mortality on ADRs. And search of literature, systematic review, and grading the evidence and develop recommendations. So this is a brief about development of guidelines in any disease, or maybe in COVID also. Thank you very much. So in case there is any question, then I'll happy to answer maybe later on or now. Thank you.
Thank you so much, Dr. Avtar, for your presentation. We will be taking the questions at the end. The next presentation, I would like to invite Dr. Sanjay Wadhwa, who's the Professor and Head, Department of Physical Medicine and Rehabilitation at AIIMS, New Delhi. He'll be presenting on the promoting physical activity during COVID-19 pandemic. Welcome, Dr. Sanjay.
Thank you very much. It is a pleasure being a part of this wonderful webinar. I will be speaking about promoting physical activity during COVID-19. Am I audible properly and my slide is visible clearly? Hello?
Yes, sir.
Thank you.
Yeah.
Actually, in these online meetings, it is mandatory to ascertain this thing.
Yes.
Yeah. So I will begin my presentation further. Now, many a times people I have seen in the medical profession, and what to talk of lay people, there is a tendency to use the term physical activity and exercise as synonyms. But scientifically speaking, it is important to differentiate. I will not devote much time to this, but this slide I think is important, even if you know, just for the revision sake. According to the American College of Sports Medicine and the American Heart Association, physical activity is any movement that is carried out by the muscles that require energy. In other words, it is any movement a person does. Exercise is by definition planned, structured, repetitive, and intentional movement. So exercise is intended to improve or maintain physical fitness. There are many recommendations for fitness and exercise in general, and for certain specific disease conditions. Non-communicable diseases are very common group for which there are exercise recommendations. And in the present times, there are certain exercise recommendations with regard to persons who are recovering from COVID-19. Some of the important sources of these guidelines or recommendations include the American College of Sports Medicine, American Heart Association, American Stroke Association, Center for Disease Control and Prevention, World Health Organization. And recently there is a new addition under the Fit India campaign. Now, there are many claims regarding the benefits of exercises, but there is moderate or strong evidence for some of the benefits. For children, there is improvement of the bone health, cognitive function, cardiovascular fitness, muscle fitness, weight status improves, and there is less chance of depression. In adults, there is all-cause mortality reduction, reduction in the stroke and heart disease, hypertension, type 2 diabetes. There are 8 cancers which are known to have a strong association with regular exercise in terms of reduced prevalence and incidence and better recovery, even if the person is on chemotherapy, etc. Depression, cognitive function, dementia, quality of life, sleep quality, reduced anxiety, depression, and improved weight status. Why weight status is mentioned everywhere is basically because obesity has become a very important health problem. And in older adults, there is reduced tendency to falls. And if one falls, if the person was doing regular exercises and physical activities, then getting up after a fall is much more likely. There is a significant benefit in people who are suffering from frailty, and overall physical function improves. Now, because the focus of the present webinar is related to dose optimization, and my previous learned speakers have mentioned a lot about the development of guidelines, dose optimization, what is the significant and all. For exercise also, there is a dosage concept. And this is governed by the FIT principle because we recommend exercises to improve the level of fitness. And in this FIT principle, F stands for frequency. How many times a day the exercise has to be done, up to what level, that is intensity, for how long, that is a time, and type, that means whether it has to be an aerobic exercise, stretching, strengthening, endurance, balance, or coordination, etc. But there are 2 more alphabets which have been added recently to make it fitter. One is evaluation of what is the effect of this kind of exercise schedule or structure, so that we can determine what is the safe rate of progression. Because if one is doing an exercise at a certain level, then there is plateau effect. Further improvement or health benefit is not so much there. So it is important to decide. And this is very important in people who are recovering from COVID-19. If they were earlier physically active, then we have to have a different program. If they were very sedentary, then the guidelines have to be considerate of the previous health status and level of physical activity also. So how much physical activity should you do? According to these guidelines, which I have just now mentioned, adults should aim for at least 150 minutes of moderate intensity activity in bouts of 10 minutes or more each week. This can also be achieved by 75 minutes of vigorous activity across the week, or a mixture of moderate and vigorous activity. All adults should undertake muscle strengthening activities such as weight training, yoga, or carrying out heavy domestic day-to-day activity related objects at least 2 days a week. And another important addition to these recommendations recently has been minimize the amount of time spent sedentary, sitting for extended periods. Intensity of exercise is classified as sedentary, light, moderate, vigorous, very vigorous, depending upon how much is the increase in the heart rate, respiratory rate, and how much is the oxygen consumption while performing that activity. So as I told you, exercises are classified as light, moderate, or vigorous. Moderate intensity activities are those in which the heart rate and breathing are raised, but it is possible to speak comfortably. Vigorous intensity activities are those in which the heart rate is higher, breathing is heavier, and conversation is harder. Many times, an average physician also cannot make out. But there are certain other scales also which are validated, such as the Borg scale of perceived rate of exertion. Now coming to the important question, should one exercise during COVID-19 times? Now, compared to other times, there are certain significant changes in the exercise related area. Pulse oximeter has become a very important object while deciding and guiding a person about the exercises, because oxygen saturation level monitoring has become very important component of the overall monitoring and treatment of COVID-19 person, because lungs are primary targets of involvement of coronavirus infection. And if the oximeter reading shows that SpO2 levels are below 94, then patients in the home care settings are advised to lie prone on their stomachs this is found to increase the oxygen saturation levels. And there are various pictures, charts, and guidelines, and even videos to train the people how to take the benefit of this active proning. The other things are certain other significant features of the COVID-19 times, and these are the mask, sanitizers, the gloves, and incentive spirometer type of equipments which are beneficial for improving the breathing capacity. When one is likely to engage in outdoor activities, then good quality footwear, and for strength building, lightweight dumbbells. Breathing exercises are very important so that if there are secretions in the lungs, it is possible to bring them out more effectively. But whatever recommendations that we are going to make, it is very important to take into account the health status of the individual. The person has not to think of competing against anybody else. The focus should be on how to improve the level of fitness compared to the previous week or a fortnight ago. Now, with regard to return to exercise in a person who is non-athlete in the COVID-19 scenario, safety advice is extremely important. One should stop exercise if any of the following occur, such as oxygen saturation levels fall below 96%, when there is no history of lung disease, new onset shortness of breath or chest pain, tightness, pressure sensation. There is a feeling of syncope or drop in the blood pressure upon activity, drop in oxygen saturations on exertion more than 3%, and symptoms of myocardial injury, myocarditis, or thromboembolic disease. Because in this COVID, it has been found that people with comorbidities are more likely to have thromboembolic phenomena, and cardiac involvement is also quite common. Exclusion criteria are hospitalized patients, patients bedbound during the infection when the infection was moderate and severe, or patients requiring cardiac respiratory evaluation prior to starting the exercises. And constant monitoring of the person is important to judge the progression. And if a patient fails to progress, then return back to a stage and seek medical review. It is very essential. Now, there are certain tips for staying healthy during the COVID-19 pandemic times. Try to do some physical activity on most, if not all days. Try to break up long periods of sitting with a regular movement program. Mix up the workout so that you don't get bored and lose motivation. Many people are enthusiastic in the beginning, but they lose steam midway and stop exercising. So that should be discouraged. And there are ways to help the person stay motivated over a longer period of time. It is important also to understand that some benefits arise with very small increase in the level of physical activity for people who are relatively inactive. And even if the person is far from meeting the recommended weekly activity levels, say of 150 minutes of vigorous exercise, etc., those first small steps are important because the benefits of regular exercise and physical activity are additive or cumulative. Aerobic activities should be undertaken in bouts of at least 10 minutes for health-related benefits. And they should ideally be performed on 5 or more days a week. And after clearance, people with recovery from the COVID-19 can engage in regular aerobic activities also. Of course, the monitoring has to be closer and more frequent. Sudden burst of activity without assessing the health status can be sometimes detrimental and fatal. In the media, there have been some examples of people who tried to be more active. They started hitting the gym more quickly. They were more ambitious and they wanted to get fit soon. They have had cardiac events and other adverse health outcomes. The guidelines for physical activity are essentially encouraged and spread by WHO and such global bodies. And taking a cue from that, in India also, under the Fit India campaign, there are fitness protocols and guidelines developed for various age groups. And this is the set of guidelines for 5 to 18 years. These are guidelines for 18 plus to 65 years age group. And these are the fitness protocols and guidelines for 65 plus years. And these are freely available in the public domain. And I would suggest people who are likely to engage in making recommendations for people in general, they should refer to some of these guidelines. Exercise is the best medicine. Unfortunately, it is not available in the form of a tablet or a capsule. Otherwise, it will be the hottest selling medicine. It is a poly pill, basically. By poly pill, I mean that it not only helps you maintain your weight near the ideal, it keeps your blood sugar levels under check, cholesterol levels maintained and low. Blood pressure is also better controlled. The chances of stroke and coronary attacks are also reduced. Osteoporosis is also less likely. Falls are also less likely. Your mental health also improves. So there is no single medicine which can have so many benefits across various systems and organs of the body. But we don't value it unless we lose it. Benefits of exercise, including walking and running, are well known to all, but it has to be emphasized again and again because people tend to take it for granted that exercise is beneficial. We know, but do we do? That is important. And I admire Professor J.S. Thakur because invariably in all his training programs, there is a session to break up the monotony of sitting posture. And I respect him for that. This is something new which I observed many years ago when he involved me in one of his training programs. There are so many guidelines and advice and recommendations how to be more active at the place of work. So the common excuse that I have no time, I have too much of work, this is not right. There are so many simple ways in which one can remain active even at the place of work. With this, I would like to conclude and I will welcome any questions. Thank you.
Thank you so much, sir. We have some questions in our chat box.
Okay.
The first question, which is on which factors become the rapid decisive factors for NCD dose optimization among the COVID-19 patients who are suffering from NCDs? This question was posed for Dr. Nusrat Shafiq.
Yeah, thank you, Ria. I think I'll take this question, and if you permit, I'll take the next question as well because Dr. Vikram had to excuse himself. So,
Sure.
So a whole lot of diseases would need to be taken into consideration for dose optimization. When we talk about dose optimization, we have to take into consideration that we will need to take care of the doses of agents which are directed towards management of COVID-19 and also the agents which a patient would have been taking for his or her comorbidity that would have been there. So the ones that would come to my mind upfront would be CKD, chronic kidney diseases. The other would be conditions affecting the respiratory system. That would again be very important, especially COPD and bronchial asthma. And a lot of our patients of ischemic heart diseases would be on antiplatelet medications and also anticoagulants. So there we need to do a lot of work to understand what would be the best doses, both for their ongoing medications and also for COVID medications. And diabetes definitely would be another condition where insulin doses would need to be modified. Even the oral hypoglycemic agents may need to be modified, especially if a patient has had the requirement of corticosteroids. But I don't think we can restrict to one or two conditions. I mean, it would be a whole lot of conditions for which you would need to take this into consideration. Again, anything which would require immunosuppressants, there again, the patient's primary drugs as well as the drugs for COVID medication for management of COVID would need to be taken care of. Yeah, so that would be briefly summarizing the response to the query which was raised. And so I think that was one thing. And there is another question which is directed towards factors becoming rapid decisive factors. I mean, factors which would be rapid decisive factors for NCT dose optimization. Again, it would be something on similar lines. One would be glucose control. That would be one rapid disease factor in patients who are diabetics or who become diabetic while having encountered COVID infection. Second would be anticoagulants. The dose would need to be modified. And third would be creatinine clearance, comes very up on my list. Yeah, I think, and the weight, of course. In fact, we have had a lot of observational data to suggest that patients with who are on the higher side of weight may not have as good as a outcome as in other weight categories.
Patients with NCDs are more or less lacking about dose optimization. So it is a good area for research, but unfortunately, COVID patients are reducing. But you really need to collect data on these drugs in patients and then do whatever modeling and those things that people in the code consortium are doing, and that would give you more definitive answers.
Yeah, exactly. So we need data for coming to a definitive conclusion. Yes, sir.
Thank you, Dr. Nusrat. I would like Dr. Sameer to highlight the questions which are there for our panel.
Can you read them, because my net connection is saying unstable? So
Okay, I would do that, sir. The next question is that can sustained physical activity affect need to go for dose optimization for NCD treatment among those who contract COVID?
Would you like me to address that or somebody else?
Yes, Dr. Sanjay. And there are two more questions.
Yes.
Should I read them out right now?
No, I am able to read in. Should
Okay.
I go ahead with the answer to all these three one by one?
Yes,
Yeah.
sir.
So for the first question, can sustained physical activity affect the need to go for dose optimization? I think the answer is yes, but one has to understand that the prescription for physical activity needs to be as careful as one is careful while prescribing anti-diabetic drugs or anti-hypertensive drugs or other medicines for treating the other non-communicable diseases. So simply writing on the card regular physical activity without actually telling in detail or demonstrating what we mean by that and how the person can judge what is moderate physical activity sometimes does not work, because many a times people who are suffering from non-communicable diseases, especially those with the cardiovascular diseases, they are afraid of exercising lest the blood pressure shoots up, or they have, because people know that some people have suffered acute myocardial infarctions after a bout of physical activity. So although the instances of death after regular exercises is very, very low, but some of these things get disproportionately highlighted in the media. So while prescribing medicines for control of diabetes or coronary artery disease, or as a person with follow-up for the stroke, if 5 minutes at least are spent explaining to the person how to begin the exercise in a safe way, how to do self-monitoring as one progresses with the exercise schedule, what are the warning signs which indicate that the exercise performance should be stopped, whether it is dizziness or heaviness in the chest, breathlessness, etc. So these things have to be conveyed to the person. And then gradual monitoring of the progression. There is a question that how to monitor the physical activity in Indian clinical and at-home settings. It is not so difficult if one understands the basic concepts. For example, everybody can have a step counter free app in the mobile phones. There are wrist watches. One can keep a track of how much of activity one has engaged in terms of number of steps. In the other exercise assistance equipments, for example, if it is an exercise cycle or a treadmill, there are ways to measure different parameters. For example, the time for which you have exercised on the cycle or treadmill, the speed of the movement of the wheel, the resistance to the wheel movement, how many calories one has burned. So one has to ideally maintain a physical activity or exercise diary in order to safely monitor the progress and decide the rate of progression. It is possible in low income settings and in Indian clinical scenario also. The third is that we should develop Indian guidelines for Indian patients. Yes, it is very essential, but there is already some base and basis for these things. Fit India campaign has, as I told you, exercise protocols. It not only includes the exercises, but also sports related instructions and the Indian component of yoga. It stresses that we should have a mix of all these three. So there is a need to develop more and move on, but we have some basis and we can utilize that. I'll be very glad to associate with any such venture. Thank you.
Thank you, sir. Now we have certain more questions, but owing to the paucity of time and we have already overshot our time for today's webinar, I would like to call Dr. Sameer to kindly conclude the session and for his concluding remarks.
Yes, thank you. I think we had a very interesting session and, a diverse range of topics. I'm sure the dose optimization concepts are maybe slightly difficult for a start, but these concepts are important and need to be understood for, everyone needs to do that. Guideline session was, I think, more understandable to everyone because you have been involved in these things, but still there were several points which were of great interest, at least to me, which we pay less attention than we should be paying. And of course, exercises, it's the best end to this session. And I thank you all. If Dr. Thakur has something to say, welcome. Otherwise, we can end this session.
It's all right. Thank you very much.
Thank you.
Thank you, everyone.